Okay, so we should be recording now. Thank you very much for reminding me. So we should have two slides from the nutrient auger plate. And so remember that if it's nutrient auger, this one pretty much can grow everything. So nutrient auger is really great for growing everything. But some of the things that it'll be really good at is doing bacteria. So a lot of bacteria samples will come from this. So if we just look at what these different samples are, we can take a look. And here we have, in this particular slide here, we have Bacillus megaterium. So this is Bacillus megaterium. So Bacillus megaterium is a rod. So this is a rod. It's a bacterium. You can't tell what the gram stain would be. We wouldn't know whether it's gram positive or gram negative because the actual simple stain, and remember we did a simple stain and we did it with methylene blue. So that is the actual stain that we used, but that's not going to tell us whether it's gram positive or gram negative. All we really know is that it's a rod and that it's a fairly long rod because that's typical of Bacillus megaterium. Down here, we can see that we have cocci. So we can see individual cells, and these are spheres. And since they're spheres, we know that these are cocci. And if we take a look that they aggregate in sort of grape-like clusters. So there's grape-like clusters of cells. And we know that that is typically going to be a staphylococcus. So because that is, typically what we see from staphylococcus. That staphylo refers to the way that the cells cluster, and the coccus refers to the fact that the individual cells are spheres. So grape-like clusters of cells that are actually spheres, we know that the actual genus is going to be Staphylococcus. That one in particular, I know is Staphylococcus aureus. So that one is Staph aureus. And then, oops, I think I have these in the wrong order. And this one here, these small rods, this is actually Bacillus subtilis. So small rods, that's Bacillus subtilis. Okay, so that is typical of simple staining using methylene blue on those three organisms. Okay, any questions about that? Okay, so there were two other plates two other slides that we should have had, and these two slides actually were taken from Sauberod's auger. And so what we can see in this slide is this looks to be pretty much, this is probably some sort of Saccharomyces. So this is yeast. And that's typical of yeast. I mean, it kind of looks maybe a little like a sphere, but then it kind of looks sometimes like it's a rod. But in fact, these are sort of oval shaped. And then, we typically can sometimes see little buds that come off of them. So we know that that's actually a yeast. Okay, then we can take a look at this sample. And in fact, this is a mold. So it has structures that are very specific, and it has these hyphae that are specific for molds and fungi. And it also has these structures that are the blastopore. And so this one in particular was actually from back some years ago, 2013, 2014, there was a mold contaminant in yogurt. And the yogurt that was contaminated was the Chobani. Has anybody ever heard of Chobani yogurt? So Chobani yogurt was contaminated with this particular type of mold, and this mold is called mucor, and the species of it is Cercinaloides. So Cercinaloides, Mucor cercinaloides. So that is the mold contaminant that was found in Chobani yogurt somewhere around 2013, 2014. So there was a big FDA alert. You know, they had to get Chobani yogurt off the shelves because they had this mold in it that was sickening some people. So not good. And so this one down here, this is the yeast. So this is also yeast, but this is the yeast that causes sort of vaginitis. And so this is sort of a low magnification, and this one's a higher magnification. Again, just doing a simple stain, methylene blue. And what we have here is the bacterium the yeast known as Candida albicans. So this is yeast, and it is the yeast known as Candida albicans. And what we can see is that these have, the germinal pore, the germinal tube, and the actual blastopore. Okay. So those are slides that, we would expect to see in typical of what we might find sometimes in the lab. Probably not this one, probably not the Candida albicans, but it might be that we would find the Saccharomyces cerevisiae, because I actually use Saccharomyces cerevisiae in other experiments in that lab. So it wouldn't be all that unusual if we had some of that floating around in the air. Might be a little weird to get this particular mold contaminant. Interesting, one of the things that actually came into the news sort of recently is that there is an organism, Candida auris, that this one has always been a hospital contaminant. So again, it's a yeast, and it's been known to be in hospitals, and they have never found it out in nature. So they've never found it out in nature until just recently. There was a research paper written that Candida auris was actually found in I believe an island off of the Indian Ocean, or out in the Indian Ocean. So kind of interesting. Alrighty, any questions about those? Okay. One thing I do want to just mention is, if you can see this was about 100x magnification for this particular Candida albicans sample, and this is about about 10 times more, about 1,000x. So it's always important to have that kind of information so that anybody viewing the actual slide or the photomicrograph would have some idea of the sort of expectations for size. Okay, so we're going to move on to the Gram stain. And the Gram stain was created by Hans Christian Gram, as we discussed in lecture, to distinguish the different types of organisms that cause pneumonia. He was really hired by a physician who was looking at pneumonia and studying what caused a pneumonia and deaths of pneumonia from pneumonia in his patients. So he really gave Hans Christian Gram free rein to study the lung tissue and develop a stain to determine the different types of organisms that could potentially cause pneumonia in his patients. So the qualities that Gram staining used to distinguish bacteria really created the two different classes of bacteria that we still use today, Gram positive versus Gram negative. So, he inadvertently did that in that attempt to look at the differences between causative agents of pneumonia. Gram staining properties also help to determine what medications might be used to treat individuals with different bacterial infections. So, it's really important to know whether an individual has a Gram positive infection versus a Gram negative infection, because the actual drugs that you would use on a Gram positive infection might not be the same that you would use on a Gram negative infection. So pretty much we're going to use this to identify between Gram positive cells and Gram negative cells. The Gram positive bacteria have a thick cell wall of peptidoglycan, where Gram negative bacteria have a thin cell wall of peptidoglycan that is actually surrounded by a cell membrane. So these cell envelope differences result in differential staining and differential response to antibiotics. Often, the antibiotics that are effective on Gram positive bacteria are not effective on Gram negative bacteria, and vice versa. So for this reason, it's important in many cases to, at the very minimum, determine whether an infection is due to a Gram positive or Gram negative type of bacteria. So we've seen this a lot. Just a reminder that the Gram positive cells have this very thick cell wall, and that cell wall is peptidoglycan. On the other hand, the Gram negative cells are going to have a thin cell wall, and that too is made of peptidoglycan, and it's going to have an outer membrane. Okay, so pretty much with this particular technique, first you would make a smear. So that's the first thing that you have to do, is you have to make a smear. And, when we say make a smear, it's to go through that whole thing that we talked about last week, where, cells are smeared onto a slide. And then once you've made that, you do that heating to basically kill the cells and also affix cells to slide. So anytime we say smear, that's what that means. Then the next thing that has to happen once the slides are cool, then you do the staining. And so the first thing that you do is you apply the first primary stain, which is crystal violet. Then you would do the mordant, which is Gram's iodine. And so this is the mordant. And this is going to pretty much increase the ability for gram-positive cells to be stained with crystal violet. Then there is the decolorization step, where 95% ethanol is used to pretty much decolorize the gram-negative cells, because the mordant didn't work on them because of that outer membrane. And then you do the counter stain, which is safranin, and that will stain actually it stains both cells with pink. But the fact of the matter is that because the gram-positive cells are already stained purple, you're not going to really see this lighter pink staining. And because of that, the gram-positive cells will be purple. Oops. So the gram-positive cells will be purple, and the gram-negative cells will end up being pink. Okay. Any questions about that? Great. So pretty much these are the gram stain results. Here we have gram-positive cells. These gram-positive cells are going to be purple. And then we have gram-negative cells, and the gram-negative cells are going to be pink. Okay. So pretty much what you would do in this is you would create 5 slides containing heat-fixed smears. So the smears that you would be making, there would be 3 tubes, and pretty much in these tubes I would have the following. I would have one tube would be gram-positive, another tube would be gram-negative, and another tube would be gram-positive and gram-negative. So you would be making smears from each of the tubes. So this would be a liquid culture, so you wouldn't have to put a drop of water on first. You would just put, 1 to 2 loopfuls of bacteria per slide. Then you would label the slides, on the left-hand side. So label the slides. Then you would have, your target area, and then you would basically put into that target area either gram-positive, gram-negative, or the combination of gram-positive and gram-negative. And then pretty much you would carry out the gram stain protocol. And it's a little bit easier to just do it one by one because there's not much time in between them. If there were 2 or 3 minutes between each step, it would be okay. But in fact, it's not that way. So the way you do it is you would begin to stain the first slide, add 1 drop of crystal violet, and stain for only 20 to 25 seconds. Then you would hold the slide, at that 45-degree angle, that we talked about before. And, you have your clothespin, and you would hold this over a beaker, and then you would essentially, aim your stream of water just above your target area. And then you wash the slide gently until the water draining from the slide is clear, which pretty much takes only about 2 seconds. It's really quick. And then you blot the slide to remove excess water. When you blot the slide, you know, you want to make sure you do not touch the target area. So it's really critical. You just remove any excess water, but you don't want to touch any target area. Okay. Any questions about that? Then the next thing you would do is you would add a drop of the Graham's iodine. And the Graham's iodine would then allow to sit on the slide to interact with the slide for about 45 to 60 seconds. After the time lapses, then you hold the slide again, you know, at that 45-degree angle, you know, over the beaker. And, you know, again, targeting the actual water stream above that target area. So after the time is lapsed, you would decolorize the slide with 95% ethanol for about 5 to 10 seconds. And that actually, you pretty much can do, you know, just follow that up, because you're just going to do that sort of right after you do the Graham's iodine. So that alcohol, you're going to just, you know, treat it. So if this is the ethanol, you're just going to treat that right afterwards. And then after that, you would then follow up with briefly washing the slide with water again, about 2 to 5 seconds. Then you're going to blot the slide to remove any excess water, as we discussed before. Finally, the last step is to add 1 drop of safranin to the smear and stain for 1 minute. So that's your longest step. After the time has lapsed, you hold the slide again at the 45-degree angle and rinse the slide with water to remove any remaining stain. Finally, you blot the slide dry, again, being careful not to touch the target area, and then just allow the actual slides to air dry for about 5 to 10 minutes. Then you place them all in slide boxes that would have been identified. Okay, so pretty much you would then view the Graham stained slides and video. You would do that actually the following week. We always allow them to completely dry, because if they're not completely dried, they look funny under the microscope. So that's why we always sort of leave a week. We don't usually look at them the same day, because sometimes they're not very well dried, because they do have to completely air dry. So that's why we always push that viewing thing off to the following week. Okay, if you do want more information on this technique, please view the video that there's the link for. But otherwise, you should be pretty well prepared to know how to do a Graham stain. Any questions? All right, well, thanks very much. Please, please, please study this, basically study these, watch the video if you would like. I'll post this recording up on our website, Moodle, as soon as possible, as soon as it's available from Zoom. But please, if you can, take the quiz. The quiz should be already available to you as soon as, I think it opens at 3:30. Please take the quiz, you know, if you can, you know, before 5 p.m. today, because I do think that leaving it till, you know, Friday, you might forget. So please don't forget. Okay, any questions? All right, thanks everyone. I hope you have a great afternoon, and I look forward to seeing you either tomorrow, Wednesday in lecture, or on Thursday in lecture. Thanks again. Bye-bye.
Thank you.
Thank you. Bye.